Thank you very much indeed, and good evening, ladies and gentlemen. I'm very pleased to be speaking to you in the home of Europe's largest cancer centre. The Christie Hospital, based just down the road, has actually got the largest number of cancer patients in the whole of Europe. And I've just spent the day there today before coming here, as they've been a long-term key opinion leader working with AGL in our Parsortix system. So we've come to the right place for this event. And I'm going to tell you about a very, very simple approach which is going to revolutionize the treatment of cancer by bringing what's called precision medicine to the treatment of cancer patients. And the issue that we're trying to deal with here, of course, is that the incidence of cancer is expanding, and unfortunately, I regret to tell you that this slide is actually out of date. Cancer Research UK have now advised us that we've got a 1 in 2 lifetime risk of cancer, everybody in this room. Obviously, partially that's because we're living longer and you have to die of something, but nevertheless, it is very concerning. And the other thing that is absolutely crucial, and this isn't well understood, is that each individual patient's cancer is different from any other patient. So if you have 3 or 4 people with colorectal cancer, you would imagine they have the same disease. Well, they don't. They have a disease in the same location in their body, but it's a completely different thing. Their disease is mutating in a different way, and it is susceptible to different drugs. And therefore, if you use a one-size-fits-all approach, which is what has been done historically in cancer treatment, that doesn't work, unfortunately. Now, we are offering part of the solution to provide personalised cancer care. Now, the benefit of personalised cancer care, of course, is going to be improved outcomes for patients. But not just that. It's also a substantial reduction in healthcare costs, because every time you give a drug to a patient that doesn't work, not only does it damage the patient, it doesn't help them, it damages them, gives them toxic side effects. And on occasion, patients die from the side effects of the drugs. But not only that, the drug is typically costing between £50,000 and some of the newer drugs up to £150,000 for a course. And that money is totally wasted. So by being much more selective about which drugs are given to which patients, you can not only improve the outcomes for the patient, avoid unnecessary suffering from side effects that are not needed, but you can also reduce the healthcare costs. And as a result, the pharmaceutical companies are actually very much focused on this. And here are 3 examples of specialized drugs for particular cancers which are only prescribed when the patient has a particular mutation. So the colorectal one there is talking about something called KRAS, and there's a drug called Erbitux, which is sold by Merck Serono. So in that case, the colorectal cancer patient will have a biopsy, and if the cancer cells are shown to be KRAS mutant, then they will be given that drug. If they're not, they won't. And that's really the issue that we're addressing. Now, the question is, how do you provide precision medicine to patients? Well, you can only do it if you actually do understand the constituents of their individual disease, their own cancer. And the way that that is done at the moment is this whole area here of the solid tumour biopsy. So breast cancer patient, this applies to all the solid tumour cancers. A breast cancer patient will have a lump. That lump will be cut out. And sometimes it's not cut out. Sometimes they use a needle to extract material, but more often than not, it will be cut out, and/or the patient may have a mastectomy to remove the entire breast. Now, what then happens is the material from that biopsy is taken into the labs, and they take out some cancer cells from inside that tumour and do lots of tests on them. And those tests identify the mutational status of the cancer and enable them to make decisions about the best care for that individual patient. And in breast cancer, for example, one of the best known drugs is Herceptin. Herceptin is prescribed to patients who are HER2 positive, so they've got an overexpression of a protein called HER2. It's a simple thing to find out in a laboratory if you have access to the cancer cell. So the patient who's HER2 positive will get Herceptin, the patient who isn't won't, and it's as simple as that. However, there is a major problem with this approach of solid biopsies. Obviously, it involves invasive surgery. Now, in the case I described of breast cancer, it is usually to the benefit of the patient to surgically remove that growth. But there are other cancers, like pancreatic cancer, for example, where it's very difficult to access the tumour to cut out the biopsy. And indeed, brain cancer, as an example, the patient may have a surgical reduction of their tumour because that's part of their treatment, but they would never have a surgical reduction simply to get cancer cells to evaluate their disease because it's far too dangerous. And in any case, repeat tumour biopsy is impossible because if you've cut it out, it's no longer there. So what happens when the cancer changes mutates and changes its disease status? The answer at the moment, unfortunately, is that you'd have no clue how to treat them. And that is why I'm sure many of us in this audience, and certainly including myself, have had friends who have had cancer, been treated, and we heard the good news that the size of their tumour has reduced. It's gone down 90%, there's only 10% left. And we all think, well, that's fantastic news. But then unfortunately, subsequently, we hear that they haven't got better, they've actually got worse, and now the drugs are no longer working. The reason for that is that the 10% that remains is actually mutationally different from the 90% that's been treated. They've had their biopsy, there is nothing that can be cut out, the disease has spread, it's metastatic, there is no site where they can go and find out which is the drug which would help that patient. And that's a terrible state of affairs, a really terrible state of affairs. So this inability to do repeat biopsies when the disease is changing, rapidly changing over time, is a key reason that people die of cancer. Now, we are seeking to address that through something which is actually conceptually very, very simple, a liquid biopsy. So by that, we mean a blood test. So everybody in this room will have had blood tests. That's what we're talking about, a simple blood test drawing a tube of blood. And the reason that that is possible is because the process of metastasis by which a primary cancer will spread is that the primary cancer will drop cancer cells in the bloodstream, and they circulate in the bloodstream. They're known as circulating tumour cells. If they land somewhere else, then they can grow there in a secondary location. That's how a secondary cancer is formed. So inside the patient's bloodstream, there will be some of these circulating tumour cells. And those tumour cells are representative of the patient's cancer, and in particularly representative of the process of metastasis, which is the growth of the secondary cancers, which is in fact the bit that normally kills the patient. It's dead simple because blood tests are easy to do. They're non-invasive. Patients don't mind having them many, many times, whereas obviously a biopsy is much more invasive. So it can be repeated. It can be done whenever you want. So if the doctors are not quite sure of the status of the patient, no problem. They can go and do another blood test. And of course, it's cost effective. So all of that is fantastically obvious. So why isn't this standard of care worldwide in treating cancer patients? The problem is a technical one. It's a technical one that my company has, we believe, solved. But that is the fact that there's only one circulating tumour cell in 1,000 million blood cells. So they're vanishingly small numbers of these cells. If you get a tube of blood, which is maybe sort of a finger full of blood, that would be just about 10 millilitres of blood, there might be 40 or 50 cells inside 10 billion blood cells. So it's very, very difficult to get hold of these cells. But if you can, then that gives you a ready access to the cancer cells of the patient, which you can then test to work out how to treat them. And I'm going to go back here. So what this is this is a picture of our, I've got one here, of our consumable. I'm going to show you an animation of this, but this is the consumable. This has got 2 granted US patents. It's a piece of polycarbonate plastic, which is covered with a plastic thin film. And we have a machine that pushes blood through this cassette, and it captures the cancer cells. The way it does that is that the cancer cells are larger than the other cells. So the red blood cells are about 1 micron in diameter, the white blood cells are about 5, and the cancer cells somewhere between 10 or 12 microns up to about 25, so significantly larger. So now I can show you a little animation showing how this works. So this is the thing I just held up. It's the same size as a microscope slide. The blood comes in through that inlet, and it will go out through that outlet. And we're now zooming in to the patented step. This was invented in the United States by our subsidiary there. And here are some steps, and the red blood cells shoot over the top, the white blood cells as well. These are showing the cancer cells, the big green ones, and they're too large to get through. So they're just going through a constricted gap, but the cancer cells getting stuck there, the other cells are going through. And what you're left with is a very small number of white blood cells and the odd red blood cell, and then the cancer cells sitting alongside. So it's a very, very simple process. Now I'm going to show you what it looks like in real life. So this is going to show actual blood flowing through one of our cassettes. So you can see the red blood cells and white blood cells streaming across. There are our steps. It's going over the steps, and then it's lighter coloured here because that's a tight constriction. They're sliding through there, and there's not such a large density of the blood. That's why it doesn't look as red. And in a minute, you're going to see a captured cancer cell. But what I want you to see here is how easily the blood flows through. It's very, very simple and easy to do. And then around about here, that's a cancer cell. So that's stuck there. Notice how it's not moving, but it doesn't impede the flow of the rest of the blood. It just flows straight past, and that just keeps sitting there. And then at the end, you've got a series of these very large cells. Now, that's a 20-micron gap, so those cells are nearly 20 microns in diameter, 4 or 5 times larger than the white blood cells. Dead simple process. Now, we've automated it, so here's the machine that we use to do it. That's about the same size as a laser printer you might have in your office. I mean, the way we've put that together is sort of reasonably innovative, but there's nothing really special in here because there's some syringe pumps, there's pneumatic pumps, some valves, some tubing, but it's not that clever. It's all controlled with a little computerized system. So from the point of view, the key thing about this is from the point of view of the user. So this is a technician in a pathology lab of a hospital. It's a really simple thing to use. They take the blood test, and that is a tube of blood. So they draw the blood straight into that tube. And the only thing they need to do is to take the top off and attach it to our machine, and then hit the button. And our machine does all the rest of the work. It pushes the blood through, up a little micro-fluidic tubing here, and through this clamp. So there's one of our cassettes. It sits inside the clamp, and there's a clamp on top of the machine. So the blood flows all the way through and out into the waste here. Cancer cells are caught in there, and that's it. And then here we have a harvest valve. We change that orientation at the end of the separation process. Hit the button again, and the machine will reverse the flow and pulse out the cells. And then you've got, essentially, you've got in 200 microliters of fluid, which is a tiny amount of fluid, you've got the cancer cells with a few white blood cells. Dead simple. And then that can be taken and analyzed in the same way that you would analyze the solid biopsy. So this is an engineering solution to a very complex medical question of how do you get hold of these cells. Note the comments here about patents. We own this technology. We're the only people who can do it like this. There are lots of other companies trying to do a similar objective, but they cannot use this approach. And we're very fortunate to have got such a smart way of doing it. And then here what you see is a list of attributes for an effective liquid biopsy system. And across the top there, you can see that our system, the Parsortix system, is simple, it's low cost. It captures all types of cancer. That's crucially important. The competition use antibodies, and they are limited. They can only do certain types of cancer. They cannot do all of the different cancers. I'm going to come to ovarian cancer and pancreatic cancer later. The antibody systems simply do not work at all for them. And also, our system captures all the types of individual cells that are in the blood for a particular cancer. So during the process of metastasis, the circulating tumour cell changes from being epithelial to a mesenchymal. It's a bit complicated, but that mesenchymal cell is the one that causes a secondary cancer. These antibody systems cannot capture the mesenchymal cells, so they miss the most important cells that are needed. We can harvest easily. Now, this is crucial. This system is owned by Johnson & Johnson. That's the established market leader. They've been FDA approved for about 6 or 7 years now. Trouble is antibody-based, and they cannot harvest cells. So what they can do is capture cells, if they're epithelial-based cells, and they can count them, which is useful because it tells you the prognosis for the patient, their likelihood of overall survival, but it doesn't tell you anything about which drug's going to make them better. What you actually want to do is harvest the cells, take the cells out of the system, and test them for mutations, and then make a decision as to which drug will be more effective. And the other key thing, which is something that we sort of happened on, actually, we realized we were quite good on purity, which means that we capture less white blood cells, which are the cells you don't want, alongside of the cancer cells. And we're about 20 or 30 times purer than any other system. And I didn't realize at first how important that is, but that is, I'm going to explain later, but absolutely crucial in the ability to easily analyze the cells once they come out. So we've got a system here which has now been well developed, and we're on a clear path to commercialization. we've done all these things. We've developed a concept here. We've productized it. I've shown you a picture of the machine, etc. KOL evaluation. So KOL stands for Key Opinion Leader. So the Cancer Research UK people with the Christie Hospital I've seen today is one of our leading key opinion leaders. So we're talking about world-class cancer research centres working with our system. And we've got 10 such key opinion leaders working with the system at the moment, 5 of whom have already reported positively. I'm going to show you that. But the 10 are now working on translational research. So we know we can capture cancer cells now. We know we can analyse them. That's fine, but it's not sufficient. We want to be proving that we can use this and benefit specific cohorts of patients. And that is what these cancer centres are working on for us right now, and that's ongoing. We also need a regulatory authorization. We are approved for clinical use. That means for treating patients in Europe with a CE mark. And we're working towards being the first company in the world to be authorized by the FDA to harvest these cancer cells for analysis. And we've been doing that for over a year now. So far as we're aware, none of our competitors have even started doing that yet. And then the key thing here is we've moved into this year research use sales. So during the second half of 2015, we will be delivering sales for the first time with this product. So we've proved that it works. And we're now taking it to the big researchers and saying, if you want to use it, you're going to have to pay for it. I've got some more comments on that. And corporate deals we'll talk about as well. We've got some major focuses on clinical applications. So this is getting this used in the £8 billion market. So at the moment, we have a lead into an £8 billion market. And these are the 5 key opinion leaders, and you can see from their names that I've already mentioned Cancer Research UK. Hamburg is a leading European place in this area of research. Bart's Cancer Institute in London, University of Surrey is a tertiary referral group in south of England, and Medical University of Vienna, who are arguably the world's leaders in ovarian cancer. All of these have uniformly had positive experiences with our system and published evaluation reports explaining how well the system works. So what we're now doing, as I mentioned, is we've moved into the sales phase. And so how do you sell a product like this? Well, the first thing is you've got to understand the dynamic of the market. And the people who really want this for research use are those people who are trying to develop the new drugs. And so there are big pharmaceutical companies which are running these cancer drug trials. And we've estimated that there are 750 trials undertaken each year at the moment in phase 2, which should be using our system. So that's what we're going to be selling into. And we estimate that that can generate £100,000 of revenue each trial for Angle. And over here, you can see that we have a very high profitability in this. We've put a hell of a lot of investment and hard work and heavy lifting into developing the system. But actually, the cost of that cassette I showed you, fully loaded, is actually pretty low compared to its value. So there's a good margin in there. And what's very interesting about this is that we can maintain a high profit margin and price lower than the cost of most of our competitors. So that's a big thing. Then moving into the phase 3 cancer drug trials, there's a smaller number, 120 there, but these are much bigger trials over a longer period of time with more patients, and each with the potential of generating £750,000 of revenue for Angle. Now, so we estimate that if we take 5% of this available research use market, that's an £8 million revenue potential for us, which is roughly speaking double what our current cost base is. So even with all the investment we're making in bringing this product to market, that research use sales potential is a very high opportunity for us. However, what we're really interested in is the sales for clinical use. That's why we set up this business in the first place, which we want to help patients have a better outcome. We want to provide a simple blood test which can dramatically improve their treatment. And as I said, we've got multiple key opinion leaders who are working with us to do that. The size of that market is very, very large. That's not a number plucked out of the air that we might get. We could only really address about a tenth of it or something. If we got 100% of the market, our revenue line would be £8 billion. That is the value of the sales that we could make. So the way I look at it, even if we only get a slither of that, we can very rapidly become an extremely large company. To achieve that, though, we need 2 things. So I mentioned regulatory authorization, but the most important thing is patient data. And that is we need world-class research centers doing extensive clinical trials with our system and proving that they can improve the outcome for their patients. And that's what all these patient studies are about. Bearing in mind we've got multiple key opinion leaders working on different types of cancer for us, we actually need to tell you about the first one that's come through. So Medical University of Vienna has done the first study that's published using Angel's Parsortix system, and they described it, and this is very unusual for an academic to speak in these terms, but as being sensational. Professor Robert Zeilinger at Medical University of Vienna described it as sensational. The reason is he spent 20 years of his life working in ovarian cancer. It's called the silent killer because it's a late, it's usually diagnosed very late. And if it's diagnosed at stage 4, the patient has only 3% chance of living for 5 years. If it's diagnosed at stage 1, they have 90%. So that's the importance of getting this right. Anyway, Zeilinger's team have basically developed over the last 20 years some markers which can accurately discriminate ovarian cancer cells from other cells, but they couldn't get hold of them. And they worked with every CTC, circulating tumour system they could, and the best they've ever got is 24.5%. With our system, they got 90% sensitivity, so 9 out of 10 women who were presenting with abnormal pelvic masses for operation, they could identify with our system whether they had ovarian cancer or not. And that makes a huge difference, because if they've got ovarian cancer, they need a specialist cancer surgeon to do their operation. And that's about 11% of the total. The remaining 89%, a general surgeon will work fine for them. So it makes a huge difference to their outcome. So we have, off the back of this, we've moved immediately to doing a large-scale patient study, which will be starting soon, together with Vienna and multiple other centres through Europe and multiple centres in the United States, with a view that in 18 months' time we will have results which will enable us to provide a unique product to help triage ovarian cancer patients and make sure they get the right treatment. And that in itself, just that individual specific application of our platform is worth 300 million a year to us. So it's an absolutely crucial area for us to focus. I'm going to summarise now, and then Ian will come and join me. But you might as well come out now, Ian. He's going to come and join me for questions. The ovarian cancer is the first area that we've got patient study results on. We've also got a lot of work going with metastatic breast cancer with the University of Southern California. We've got, down the street here, we've got work with pancreatic cancer in Manchester and also colorectal cancer. We've got work with Bart's Cancer Institute in prostate cancer. So there are multiple other clinical applications which are coming through. So I think what I'm going to do is leave that slide up and then take questions.
Thank you, Andrew. Questions, anybody? Gentleman just there.
Hi, I'm wondering to what extent you're likely to get piracy on your equipment. You said it's a very simple machine to build. The cassette, once someone's got hold of an original, it looks relatively simple to copy. How would you go about stopping that happening if you've got all the patents?
Yes, so in terms of the ability for somebody to mimic the product, the cassette that I held up is built to plus or minus 1 micron, so there is a lot of precision involved, and there are only a relatively small number of companies who could actually make such a device. The one we use is Thinks in Germany. But the answer to your question was within the question. It is the granted US patents that we have which will protect anybody else using this. There are other people trying to do other ways of capturing the cells, but at the moment they are suffering substantial problems, like they can't get the purity, they can't get the cells out for analysis, etc. I don't believe that we'll be the only solution to this, because there are multiple, I mean, there's probably 20 companies working as hard as they can on this, but it's a large market, and so far we've found in the ovarian cancer we're the only system that will work on that. We heard today that in pancreatic cancer, Manchester thinks we've got a number of very key attributes. So I'm accepting of other people doing it different ways. They cannot do it our way because we own that.
Next question, anybody? Gentleman at the back.
I've got 2 questions. Can you give an update on FDA approval? And secondly, I understand the biggest market for cancer is China. Have you got any moves to go into China?
Should I take this as well? Okay, so the FDA, we're trying to do something which is extraordinarily difficult. We want to be the first company that's ever been authorised by the FDA to harvest the cancer cells. We're in a good dialogue with them, albeit it is what I would describe as a bit staccato. In other words, we have a conversation and then they go away and think about it for a bit and then we have another conversation. However, they have expressed their positive support, and President Obama himself has talked about the need for precision medicine in treating cancer, so the FDA is aware of the need for this. In terms of how long it's going to take to get that sorted out, it's difficult to know, because the other thing that we've done, which has challenged the FDA, is we've said we want a platform approval. So we want an approval to capture cancer cells for any kind of analytical purpose, and they find that quite hard to get their head around, because normally people go and say, we're doing this, and this is the very specific application, and here's the patient data. We're saying, no, all we want to do is part of the jigsaw. We want to capture the cancer cells and provide them in a form that can be analysed, but we don't want to prove every single analytical approach. So that's your first question. The second question was China. We've had 3 different organisations approach us about distributing product to China. At the present moment, we're avoiding it for the reason that we think there's big markets right on our doorstep, and in the United States, we've got a big push in the United States. We've hired a major person to help us do that. And that's an easier market at this moment in time. I think when we're further developed, we'd probably go for China, but not now.
And I'd probably just add, by value, we reckon the US is about 60% of the value of the market, so that's our prime market, along with certain European markets. So although China may have the numbers, if you look at the value that you're going to get paid for providing the services and pushing through the cassettes and so forth, the real value for us is in the US, so that's our primary market at the start.
You mentioned you've got CE mark. Is that for clinical use in Europe, or is that just for laboratory use?
It's cleared for use in Europe.
So you could be selling to hospitals?
Yes, yeah, we can sell to hospitals right now. The thing that's stopping us selling to hospitals in Europe now is a need for robust patient data, which has been proved out.
Right. But the FDA would follow then for the US. Once you've got the FDA approved, you can then do clinical in the US, I presume.
When we've got FDA, yes. Yeah. Yes, exactly.
Question. So as far as the analysis is concerned, have you got links with the people who do the analysis? So you provide the tumour cells?
Yeah, so our system's designed to be, the term we use is open source, so it will work with any of the analysis systems. So there's various techniques. You may have heard of next generation sequencing. We provide the material into that. There's PCR, there's FISH, there's various techniques. So we're designed to just provide an alternative form of biopsy material so that we can work with any particular analysis platform. So it's a bit like an analogy, if you're taking the gold mines, we're selling picks and shovels to all the different gold miners. So we over time, I'm sure we'll develop some collaborations, but at the moment we're designed to work with all the different analytical platforms.
Yeah, we want to be like Intel inside, Parsortix inside, so we want to be in every single analytical system.
Sata, you've got a question, I know.
Oh, well, never mind. Yes, a bit further to the previous question. Is your sample size actually big enough for people to analyze the different biomarkers, which obviously are of major importance for personalized medicine? If you only capture a couple of cells, can you do, for example, genetical expression analysis, which are very much in need?
That's exactly where the patient studies are working at the moment. Historically, the CTC systems have been, they haven't had enough ability to capture enough cells to work with all patients. So, for example, historically, they say about 25% of patients. The example I gave in ovarian cancer, we've got 90% sensitivity, and that's on the first time out. So it is looking very, very strong. What is good about it is particularly good on the RNA and protein expression, because other systems like cell-free DNA can't touch that. And so we're finding that sensitivity of our system does enable that.
And I think I'd probably add as well that some of our corporate collaborations, so there's one with the EKF Diagnostics, they have an incredibly sensitive analysis tool, and we're doing some work with them, and that would allow us to work with, in certain cases, even smaller samples.
Potential new client then.
We'll get to that.
Any other questions? Roland Scott.
You talked about the first wave of revenue coming from clinical trials and the different stages and the different values that they have the potential for. For that sales process, do you have your own sales teams out in the field, or how are you making those relationships and selling those units?
Yeah, so we're not, we do have a small business development team, but they're not involved in that kind of sales activity. What we're using is the relationships with the key opinion leaders, and actually the way that the pharmaceutical companies set up their drug trials. Somebody coming and knocking on their door and saying, would you like to use my test? That's not going to work. What is going to work is the big cancer centers who are involved in doing those trials. If they say, we've evaluated this system, we think it's an additional biomarker, you should use it, that's the way to get in. So we're using our key opinion leaders to open the doors for us.
So they then generate the conversation about the product or the solution, and then they want to come and use that as part of their development.
Yes, and what we're seeking to do here, by the way, is in terms of the drug trials, is to help the pharmaceutical companies reduce the cost of their drug trials and to accelerate the timescales. And the reason is that they need to look for an indication of the effectiveness of their drug. And typically they looked at the tumour mass size. Obviously the long-term prognosis for the patient, you know, the overall survival rates, but that takes a long time to complete a study. So they've looked as a proxy, they looked at the size of the tumour. Well, as I explained before, it could reduce by 90% and still come back. So if you actually look at you can analyse the status of the cells, the cancer cells, that actually can be a good alternative proxy marker.
Time for just one more question. Anybody have any questions at all? Gentlemen, this door's back.
Hello, yeah, I'm not a doctor, but I'm just wondering, does your equipment say anything about the stage the cancer is at? In other words,
will our system,
because that would affect the diagnosis and the treatment.
I think our system is not particularly to identify the stage, but what it can do is a number of very important things. In the ovarian cancer example, it can identify the existence of the cancer ahead of it otherwise being detected. So a particular example which has been very topical in the last week is Angelina Jolie, who has had an elective removal of her ovaries. Well, the people in Vienna think that our system could be a blood test that could be used quarterly instead of going to that kind of surgical removal and be an identifier of an early onset of disease. Now, another application is in terms of remission monitoring. So patients who have been treated, they're in remission. The question is, are they going to relapse? Well, there's a possibility that the system may give an advance notice of possible relapse.
Okay, just another point is, you mentioned the type of cancers that you're trying to use it effectively, but what about the blood cancers, which is leukemia? Have you actually looked at leukemia as capturing the cells?
So so far we have kept away from leukemia. The reason being that is obviously it's a blood cancer, and therefore there are a lot of cancer cells in the blood, and we've been primarily focused on very rare cancer cells in blood, whereas in leukemia it isn't the case. There are some issues relating to minimal residual disease in leukemia, which some of our partners have said they would like to investigate using our system.
Thank you very much, and thank you, Andrew. Thank you.